first level of the immune system is the innate immune system, which I like to think of as the non-specific or general immune system. This part of the immune system's job is just to recognize general signs that are associated with different pathogens. The two signs that we talk about are pathogen-associated molecular patterns. So those are generally proteins on the outside of pathogens that are common in all different types of bacteria or viruses, and they will bind to the toll-like receptor of a white blood cell or leukocyte, and they will cause downstream cascades that will eventually help to kill that particular pathogen. Similarly, there are damage-associated molecular patterns that occur when a cell is either infected by a pathogen or it's damaged from working out, or it's damaged from a crush injury or something like that, and that will also bind to this receptor and cause the immune system to become involved to help remedy the problem. The two particular types of white blood cells that are used by the innate immune system are phagocytes and granulocytes. Now, phagocytes are just cells that use a process called phagocytosis to destroy the pathogen. And phagocytosis is just a fancy term for basically eating or digesting a bacteria. So the bacteria or the pathogen. So the pathogen will bind to the toll-like receptors just like before. It'll be taken up into a vesicle. A lysosome will be released into this vesicle, and the lysosome will bring free radicals which will help to break down that particular pathogen and into a bunch of different components so that it can't hurt any of the cells in your body. And this part on the right here is just kind of a general overview of what happens inside when a lysosome uses their free radicals to kill that pathogen. Now, this might look very complicated. This is essentially an organic chemistry mechanism of action diagram. I'll kind of go over it, but some of you may not get it unless you've taken a chemistry class. So basically, a free radical is a molecule that has an electron that is very hungry for another electron pair. So it's very reactive. It's searching everywhere to get that extra electron. So it's able to take that electron from virtually any molecules, even molecules that are considered relatively stable under normal conditions. So in this case, this hydroxyl radical takes this hydrogen ion and an electron from an unsaturated fatty acid in the plasma membrane of a bacteria cell. And this will create a stable molecule called water. Everybody knows what that is. The problem is, even though now you've solved that problem, you've created a new problem, and that unsaturated fatty acid now has an electron that also wants to be paired up with a buddy. And this ends up getting paired up with an oxygen floating around. So now you've solved that problem of that carbon needing its extra electron, but you've just created another problem where now you have a peroxy radical with another electron that's searching for a buddy. So you can see this is basically a never-ending process of this one a lone electron wanting to be stabilized by finding a buddy, and it never quite does. And it ends up not only breaking down different molecules, but also causing them to not be able to function properly. So even if it isn't broken down, it's pretty much useless. Moving on, the other type of leukocytes present in the innate immune system are called granulocytes. And granulocytes are simply white blood cells that have vesicles or organelles inside their cell that release different molecules out of the cell to help combat pathogens. So once again, we have the DAMP or the PAMP, which is recognized by the toll-like receptor, and instead of ingesting that pathogen or bacteria or whatever, it will release molecules out of that granule. Moving further, the different phagocytes are neutrophils, macrophages, and dendritic cells. Now, neutrophils can be differentiated from the other phagocytes because this is the most common leukocyte that will be circulating, the one that a lot of lab values when you go to the hospital will focus on. They're also the only leukocyte that is both a phagocyte and a granulocyte. They use a process called chemotaxis to search out different pathogens, and what chemotaxis means is it follows a gradient of a particular molecule, kind of like breadcrumbs, to find a particular pathogen. So just like Pac-Man here, it'll follow breadcrumbs to that pathogen. And the way this works is it will flow from a low concentration of that particular molecule to a high concentration of that particular molecule, which will lead it to the pathogen. That's what chemotaxis is. Furthermore, it produces things called nets, which are neutrophil extracellular traps. And this is basically, it is able to utilize histones, proteins, and DNAs within itself to kind of wrap up different pathogens outside of the cell near it. So it does a lot of cool things. Macrophages can be differentiated because they're basically the most basic of phagocytes. They don't have a lot of other things that they do. They're not quite as cool as neutrophils. But they can also be called monocytes when they're in the bloodstream. And then when they enter into a particular tissue or site of where a pathogen is, they are then called macrophages. And then dendritic cells are phagocytes that are only present in tissue of the external environment, mainly your nasal passages. Moving on, the different granulocytes are separated by the types of nucleus that are present in the inside of their cell. So neutrophil has multi-lobe nucleus, eosinophil has a two-lobe nucleus, and a basophil has a bean-shaped nucleus. We already kind of talked about neutrophils, but eosinophils can kind of be differentiated because they're able to target organisms that are too large to be engulfed or phagocytized, like we talked about earlier. So they'll secrete things called major basic protein and eosinophil protein to help chop them up so they can either be phagocytized or they're just dysfunctional at that point. Basophils and mast cells are cells that are both generally involved in allergies and the inflammation that comes along with allergies. And they're activated by immunoglobulin E, which we will talk about later. Once they are activated by immunoglobulin E, because of some allergen that's entered the body, they release histamine and serotonin, which will cause vasodilation and kind of a warming of the tissues. And also heparin, which is an anticoagulant. So this is why you get the classic symptoms of inflammation being, swelling, hotness, and redness, because there's more blood flow because of the vasodilation and the anticoagulant, which makes sure that the blood doesn't clot there, so that more cells can flow through the area and carry out their functions. And then between basophils and mast cells, the main difference is basophils are in the bloodstream, mast cells are generally just in connective tissue and mucous membranes. Basophils can be in some tissues though.